Before going to discuss about the gram staining procedure, let's know the overall scenario of a specimen from collection to identification. With the help of gram stain, we can directly detect the bacteria in the clinical specimen, whether it is gram positive or gram negative. Then we have to perform catalase and oxidase test on all types of specimen. For gram negative organism, we have to do the common biochemical test routinely, that is indole test, site utilization test, urea hydrolysis test, triple sugar iron test. For the gram positive specimen, we have to do catalase test with the help of hydrogen peroxide. Catalase test is primarily used to differentiate between Staphylococcus and Streptococcus. Staphylococcus species are catalase positive and Streptococcus species are catalase negative. Staphylococcus species are arranged in cluster, which can be further divided into coagulase positive Staphylococcus or coagulase negative Staphylococcus with the help of coagulase test. Staphylococcus aureus is the most pathogenic species, which is coagulase positive. Other Staphylococcus species like Staphylococcus epidermidis, Staphylococcus saprophyticus, Staphylococcus hemolyticus, they do not produce coagulase, so they are coagulase negative or cons. Another test named as the Novovisin test helps in differentiation between Staphylococcus epidermidis and Staphylococcus saprophyticus. The Staphylococcus epidermidis is susceptible to Novovisin, where the Staphylococcus saprophyticus is resistant to Novovisin. Staphylococcus saprophyticus primarily associated with urinary tract infection, particularly in young women, while Staphylococcus epidermidis is more commonly linked to infection related to medical implants. In the next video, we are going to discuss about the gram staining procedure with all related questions and answers.